Throughout our lives, we are exposed to a wide range of chemicals. In order to minimize the risk of these chemicals, risk assessment is required by law. At present, such risk assessment is conducted using conventional toxicological methods based on data from animal experiments, which currently represents the regulatory standard. However, in recent years, a paradigm shift has been underway, away from animal testing towards a safer, more effective and more sustainable risk assessment. At Fraunhofer ITEM, scientists are actively shaping this paradigm shift. With many years of expertise in the field of toxicology and risk assessment, they are developing new assessment strategies, so-called next-generation risk assessment. The researchers use experimentally obtained data from in vitro and ex vivo models, also developed at Fraunhofer ITEM, as well as computer-based in silico methods. These new methodological approaches are referred to as New Approach Methodologies, or NAMs for short, which comprise a broad spectrum of animal-free approaches and methods that help to better predict the safety of chemicals for humans. To this end, scientists use human cell cultures in vitro or human lung tissue samples ex vivo. Are the chemical structures similar? Are the toxicological effects comparable? To find out, the researchers at Fraunhofer used ToxTool, a compilation of various toxicological databases that they set up for this purpose. The data stored in these databases on analogous, i.e. similar structures, can be analyzed using read-across methods or structure-activity relationships. NAMs also include data-based evaluations, i.e. in silico models. Existing data on chemicals is used to assess the potential risk of a new chemical. An innovative approach to the risk assessment of chemicals is to observe the adverse effects of a substance in the organism, the so-called adverse outcome pathway. A chemical triggers an initial event in the organism, which in turn causes further key events that lead to the toxic effect. To assess whether a chemical has a toxic effect, tests are conducted at various stages to see whether the chemical takes the toxic adverse pathway. This method does not involve animal testing. AOPs can help reduce or even eliminate the need for animal testing. But what dose of a chemical is relevant for humans? How can in vitro test results be extrapolated to the in vivo situation, in animal models or directly to humans? To answer such questions, the IVIVE approach uses PBPK models to extrapolate from in vitro to in vivo. Doses are derived from in vitro and in silico data using mathematical modeling. Various case studies have shown that this type of risk assessment works. Fraunhofer ITEM uses its expertise to develop and consistently advance such innovative approaches to risk assessment. The Institute's goal is to incorporate these new methods into binding regulations in the long term. In this endeavor, the Fraunhofer researchers are not alone. They collaborate closely with partners from industry, science, and government agencies in EU projects to promote the recognition and implementation of a next-generation risk assessment.